Hello, biology students. I'm going to just pick up with notes kind of where we left off in class. I think we went through this slide in talking about the different levels of structure that we see in proteins. So the primary structure is just the simply those amino acids connected together, but there's not really much for useful structure there. We do not see proteins remain as primary structures. There's always a greater destiny that awaits a polypeptide chain or a primary structure protein. So these are held together by those peptide bonds. And then it's going to fold in kind of a secondary structure. And I think we looked at this visual. We talked about, there's a couple of different structures they can, this string of beads, if you will, this polypeptide chain starts to take on some specific structure. It's either pleated sheets or it's this kind of helix called a alpha helix. That would be secondary protein structure. And again, most proteins don't stay in a secondary structure. There's more structure needed in order for them to become a functional protein. So then there's going to be some other folding to move that into a more complex tertiary structure or third level structure. And there is some hydrophobic and hydrophilic regions both. So I know I said hydrophobic, which is water fearing. You guys knew that. But there's also some hydrophilic that also is important. So those amino acids, remember, have different properties. And that's where we see that hydrophobic, hydrophilic, some charged ends, some ends that are polar, some that are attracted to other amino acid ends, and so forth. So all of that becomes important in folding into that tertiary structure. And that is not all. There is one more level beyond that. So the tertiary structure, you can see, so the pleated sheets are, some regions in here are of those pleated sheets and some are the alpha helix structure, but you see that a lot of folding has taken place. It's folded over on each other and so forth. And then the quaternary structure, some of those are going to bond together to make a large, more complete protein that has a very specific shape. And that structure is incredibly important to the function of the protein. So it has to fold and form exactly correctly, especially if we think of things like enzymes where it has to fit that substrate like a lock and key mechanism. So those folding rules, if you will, and processes become very, very important. So other structures that we see in proteins, something called motifs, and these are recurring structures that we see over and over and over again, really common in proteins, and also domains. Domains of proteins are kind of functional units, again, that we see in lots of different proteins. So it's more complex than just that alpha helix secondary structure or those beta pleated sheets that form that secondary structure. It's a lot more complex than that. So with this tertiary structure, we might see a domain that looks something like this, and then in a quaternary structure, so these are connected by a single polypeptide bond between the domains, but we see lots of these domains bonded together and hooked together in that quaternary structure to form that complete protein. And then there's some other kind of tricky things that happen with protein folding that are very important as well. We have something called chaperone proteins. So if a protein doesn't fold correctly, it's pretty useless. So we do have that hydrophilic and hydrophobic properties that are important kind of for the beginning folding of that protein, but that would take a very long time for proteins to fold the correct way if it were only due to those kinds of forces like hydrophobic, hydrophilic, and charged ends and attraction to other amino acids. It would be a really, really slow process. It would take way too long. So there's these chaperone proteins that sort of chaperone the folding process that then helps it fold a lot faster and ensures that it folds exactly in the correct way. So they become really important. These chaperone proteins are so important and so integral in getting that protein to fold to exactly the correct shape. And your body, your life literally depends on these proteins being folded in the correct way. Imagine the enzyme or the enzymes that broke food down, that eventually broke more complex molecules down into glucose, which needs to get in your cells. If you lacked the enzyme that broke those down, you wouldn't live very long. Insulin, which is the carrier molecule that carries glucose into your cells, if that wasn't formed properly, you could never move the glucose into your cells. You wouldn't live very long. So the exact specific shape of those proteins is so important. So these chaperone proteins just sort of ensure that that is happening both fast enough and exactly in the right way. And then denaturation of proteins. Proteins exist in a pretty narrow range, a lot of them, in a pretty narrow range of proteins and pH and ion concentration in your bodies. And we have to maintain homeostasis and regulate all of those things. So we have to have our temperature. You know, this is one of the reasons why if our temperature gets too high or our temperature gets too low, we don't do very well with that as humans and as warm-blooded animals. But there's a lot of other things too that need to be maintained. But if proteins are exposed to an environment that's, for example, too acidic or it's too basic, it actually can denature those proteins, and that means kind of break them down. So the visual down here, hitting a button, sorry, hit a button, not starting over. It takes too much time to edit. So we're just gonna go from here, pretend that never happened. So anyway, so imagine like if you take an egg, which is high protein, so an egg is really a cell, and you expose that to high temperatures, you can see literally as that egg white that was clear begins to turn white. That's exactly what's happening, is the proteins are denaturing. You are breaking those protein bonds that hold it together, and then it takes on a very different look. So we do, that's one way that we can look at that. Well, if you put acid on a raw egg, the same thing would happen. We would see a denaturing of the protein. The egg white that was clear would turn a white color. And sadly, if you, the same thing would happen to your eye, which has a clear kind of coating on it. If you got a strong acid in your eye, this is why we're so vigilant about wearing protective eyewear and chemical safety goggles in the classroom. It would do the same thing to your eye. Your eye would turn white as that protein was denatured, and it wouldn't be very functional, and it's not reversible. And again, just really quickly, we can see all the folding that is involved in this normal protein. And when it's denatured, we lose that biological activity of the protein because it doesn't work right anymore. We have broken bonds that needed to be there. The protein had to be a specific shape. And I like to use enzymes because they have such an incredibly specific shape, but other proteins too, they fold a certain way for a reason. And so we have denaturation. See how it's like this nice kind of ball of thread that now is sort of all loose and the structure is gone. So this is that denatured protein. This shows a renatured renaturation. A lot of proteins, once they're damaged, cannot be renatured. So it's important for us to maintain homeostasis in our bodies. That helps maintain the integrity of those proteins.